بسم الله الرحمن الرحيم. Assalamu alaikum wa rahmatullahi wa barakatuh. Thank you to our moderator for giving me the opportunity to present the results of our research on dry transportations of Oryzias celebensis embryo for ecotoxicological studies. As we know that the genus of Oryzias has long history of being used as sentinel organism for ecotoxicological studies. The reason why we use them are Oryzias fish have small size. This means that if we use the fish in the test, the study cost will be cheap, do not need a lot of workers, produce less waste, and are easy to handle. The second, the fish have a short regeneration time, so we can do a study at every phase of their life cycle in a relatively short time. The third, the fish have sexual dimorphism, so we can distinguish male and female morphologically. This makes it easier for us to choose whether we use male or female in the study. The fourth, Oryzias fish are easy to maintain and breed at laboratory level. The fifth, the fish spawn daily for 3 to 4 months in the laboratory. The coherence of the Oryzias egg is transparent, so we can document and analyze embryonic organ under microscope easily. The reason for using the embryo in ecotoxicological studies are the embryo is very small compared to the adult fish. The diameter of embryo is around 1.5 millimeters. The second, the corn is hard and transparent, facilitate non-invasive observations of the embryo throughout development. The embryo is more sensitive to toxicant compared to the adult animal. Actually, to produce this protocol in laboratory, we need to perform intercalibration between one laboratory and another. Therefore, we need to transfer specimen between laboratories. Actually, during my research on the Oryzias genus in the laboratory, we often found the eggs that had not been harvested and were attached to dry media, such as on the top of an aquarium pump. And after being observed with a microscope, the eggs were still alive. So we were inspired by this phenomenon to conduct research on the dry incubations of the embryo. Therefore, we hypothesized that the embryo of Oryzias celebensis could be dry incubated. Then we did research with the following method. We use a microplate with well volumes of 3 milliliter. There were two treatments with 4 replication. The treatment were tried with water media and treatment with embryo rearing media. The embryo were incubated for 7 days and usually the embryo hatched on the 8th day. The statistical test that we used was the student test. From the results of statistical calculations, it was known that there were significant differences in the average of incubation periods of Oryzias celebensis embryo that incubated in dry condition and embryo rearing media, as you can see in this figure. The incubation periods of dry condition faster than ERM. Probably it was due to the temperature, and the temperature in dry condition was probably hotter than in RM. However, if we see this figure, the total length of the incubated larvae had a longer total length than those reared in ERM. So it can be concluded that the embryo of Oryzias celebensis, which have hard and transparent chorion, can be transported dry for value study, especially for ecotoxicological studies. That's all our presentations, and thank you so much for your kind attention. Assalamu alaikum wa rahmatullahi wa barakatuh.